All right, let's talk a little bit more about the adaptive immune response and B and T cells. So the four characteristics of the adaptive immune system, we'll talk about each of these a little bit more. The diversity of the lymphocytes and their receptors. So this is a reference to the fact that the genes that control the variable region on the receptors get rearranged as these cells first develop during actually during fetal life and early infancy to give us the huge assortment of possible variable regions that are found in the mature immune system. Self-tolerance. Of course, if we're just randomly making a bunch of different antibodies and receptors, some of them are going to match epitopes on our own cells. So those have to be eliminated because we don't want the immune system reacting to our own proteins. So one of the things that happens during B and T cell development is an elimination of those self-reactive molecules. B and T cells proliferate after activation, and this gives us our immunological memory. And we have both memory B cells and memory T cells. So we need self-tolerance because the antigen receptors are just generated by rearranging the DNA, and just by chance, many of those, probably hundreds or thousands of them, are going to accidentally be reactive to our own cells. So those have to be eliminated, and this happens by either destroying those cells and triggering them to go through apoptosis or programmed cell death, or making them non-functional so that they won't respond if signaled to do so. They will no longer react to those signals. And this generally works. So in the lymph nodes, we have these B and T cells that are waiting to be presented with an antigen that matches their own receptors. And when they bind to an antigen that matches their receptors, that activates them. And we'll talk about helper T cells, which are an important part of this process as well. So once they get activated, our B cells or T cells start to divide. That's the first thing that happens. And this is called clonal selection. We can do this in the lab as well. And again, a reminder that that word clone just means a bunch of genetically identical individuals. And we could be talking about whole organisms. So if we say these two mice are clones, we would mean they were genetically identical. But we also could use it to talk about cells. So if we have a population of cells that are clones, we mean that they're all genetically identical and they were made by just allowing one cell to divide. And then we get a whole culture dish filled with identical cells that were made by mitosis. And so that would be clonal selection. And it happens in our bodies, and it also happens, we can do it in the lab as well. So those clonal cells, so they're all genetically identical. They all have exactly the same genes for these receptors, these antigen receptors. But as the cell divides, some of the cells are going to have different fates. Some of those cells are going to turn into plasma cells that are going to start to secrete antibodies. Other ones are going to turn into memory cells. And the memory cells don't secrete antibodies, but they sit there and they wait to see if this is encountered again. Now we know, and you probably have heard from listening to the news about coronavirus patients that have been, or COVID patients, I should say, that have been studied, their antibody production is very high a few weeks after they were exposed. And then it slowly starts to go down. And 3 or 4 months after they get well again from COVID, their antibody production, they're still making antibodies, but the antibody production is low. And people have used this as cause for alarm, but in fact, that is what always happens. That happens with everything. That low level of antibody that remains, that is what we expect for every virus that we're exposed to. And what that tells us is that there are memory cells. So those people who have recovered from COVID, they have memory cells, both B cells and T cells, that are looking for that virus again. And if that virus ends up in their body a second time, antibody production is going to be ramped up extremely rapidly the second time they're exposed to it. And that's what we want. So the fact that they keep saying, oh, antibody production goes down after 3 or 4 months. Yes, it does. That is what it does for literally every virus you're exposed to. That's normal. So here we have that demonstration of that clonal selection. So we have a bunch of B cells here sitting in the lymph node waiting to be exposed to an antigen. One of them matches an antigen that's been exposed to it from a dendritic cell, or that gets into the lymph node. That one is now activated. This B cell is now activated, and it's going to proliferate. It's going to make lots and lots of copies of itself. This is clonal selection. Now, some of those cells are going to become kind of dormant and become the memory cells. The rest are going to become plasma cells and start secreting antibodies right away. Now, eventually, these antibody secreting cells start to die off, and there's fewer of them, which is why if they don't continue to be stimulated, antibody production goes down, is what we would expect. But now, instead of having a single cell here that can respond to this antigen, now we have thousands of cells that are all going to respond if this same antigen appears again. So that is really what is happening when you've been exposed to something one time, and now you're not going to get it again. You're still going to be exposed to the virus, but you won't get sick, or bacteria, because now you have memory cells that are going to ramp up a response very quickly. So here's a little short video about that. There's quite a few videos in this section, so I'll show you a few of the short ones, but not all of them, because some of them are a bit long. This is a kind of a shorter one that's a nice little
Humoral
summary.
immunity, carried out mainly by B cells, deals with invaders in body fluids. B cells are lymphocytes that send out antibody molecules in response to certain stimuli. There are many kinds of B cells. Each kind produces and presents its own uniquely shaped antibody receptors in its plasma membrane. These unique receptors only bind to antigens with specific shapes. The B cell that successfully binds to an antigen is stimulated to proliferate into a clone of cells with the same specificity. This process of antigen-selected proliferation is known as clonal selection. B cell proliferation produces clones of plasma cells and memory cells. Memory cells are long-lived cells that can respond rapidly to future exposures to the same antigen. They account for the speed and strength of the secondary immune response that gives us long-term immunity to a disease. Plasma cells multiply rapidly. They produce and secrete antibodies in increasing amounts, peaking about 2 weeks after the activation of the original B cell. These antibodies flow through the body's fluids and tag the original foreign antigen molecules, marking them for destruction.
So monoclonal antibodies are something that we've been hearing about, and monoclonal antibodies are made in the lab, and they're made by clonal selection. So there's a few different techniques for making these, but basically they involve isolating B cells from an animal that's been exposed to whatever virus you're interested in or bacteria that you're interested in. And then all those B cells are separated and grown separately as single clonal colonies. And then the antibodies that those single clonal colonies produce, which will only be one type, can all be tested. And then if you find that one of the antibodies happens to be a neutralizing antibody for whichever virus you're interested in, now you have a population of cells that's producing that antibody, and as long as you keep growing those cells, you'll continue to be able to harvest antibodies from them. And it's called monoclonal because mono means one. So monoclonal antibodies means that we're getting an antibody of a single type. Now, this is not normally what your body does. Normally, your body, if you're infected with coronavirus, for example, you're going to produce an assortment of antibodies that all recognize slightly different epitopes on the virus, and you're going to produce all those antibodies. And maybe only one of those antibodies is a neutralizing antibody, but you're going to produce this mixture of different antibodies in response to that virus. So what this does is allows the single antibody, the best antibody, to be isolated and then grown in the lab, produced by cells that were growing in the lab, and then use that as a therapeutic. And they're even experimenting with deliberately making cells using genetic engineering techniques like CRISPR to make cells that produce the antibody that has the binding characteristics that we specifically want. So that's the basis of these monoclonal antibodies as a therapeutic. So if you hear that something is a monoclonal antibody, that's not something that was isolated from a patient that recovered from COVID. That's something that was grown in a lab using cells that were, sometimes they're a mixture, they're hybridized cells, they're mouse-human hybrid cells. So there are concerns with side effects from these kind of treatments because often the regions of the antibody, not all the regions are from our human genes. So they could have cross-reactivity or generate an allergic reaction, but very useful as therapeutics. And we do have some therapeutics for diseases that are, in fact, antibodies. And we'll talk about that at the end when we talk about passive immunity. So that's a type, giving somebody monoclonal antibodies is a type of passive immunity. So the first exposure to an antigen, when we generate the first population of memory cells, that's the primary immune response. A clone of B cells and a clone of T cells are produced as a response. The next time that same antigen is shown to the immune system, now we get what's called the secondary immune response. That means that antibodies are going to be produced much faster. The cell-mediated response is going to be much faster. Instead of taking 2 weeks, it's going to take a couple of days to get a really good response. So the red line here and this line here, this is a primary response. So here it says, on day 1 here, we're going to expose to the immune system to antigen A, whatever that is, virus A. We get our initial response, and you'll notice this is days at the bottom here. So we get peak antibody production at about 14 days. So it takes about 2 weeks for that primary immune response to ramp up a really good antibody production. That's why for most viruses, even cold viruses, you're going to feel sick for about a week if it's something mild. That's about how long it takes to go from here to here. Now, the second time we get exposed to antigen A, that immune response is going to be greater. We're going to get a higher antibody titer in the blood, and it's going to happen really fast. It's going to happen in a couple of days so that you're not going to be damaged or get sick the second time. And again, we have the first time being exposed here to antigen B, and we're going to get a primary immune response to that, which is lower level. And this is why sometimes for vaccines, they want to give you a booster shot. So this would be the first vaccine. This would be the booster shot. So they bump your antibody production up to a higher level and bump up the number of memory B cells and memory T cells that you're going to have by giving you a second dose of the antigen. That's a booster shot. So that is those are the two parts of the adaptive immunity. The humoral immune response is the antibody production. These are not cells, they are proteins that are secreted that can bind to a specific epitope. They're in your blood, they're in your interstitial fluid, they're in your lymph. Antibodies can go everywhere. The cell-mediated response, that's T cells. So, and remember, we do have other cells like natural killer cells. Those are part of the innate response. They're not responding to a specific antigen. T cells are, the cytotoxic T cells are responding to a specific antigen, and they reproduce by clonal selection. When they're activated, it's the same timeline we're looking at, 2 weeks to get a good response. There's another kind of T cell called a helper T cell that's really important in this process. Helper T cells, when they are activated, and these are in your lymphatic system, when they are activated by an antigen-presenting cell, they produce molecules that stimulate both B cells and T cells. And so they were really kind of a key point in the immune system. And you'll recognize that these are CD4, well, there are CD8 ones too, but there are CD4 helper T cells. That is specifically one of the molecules that's part of the receptor that recognizes the molecules that are being presented to them by an antigen-presenting cell, which is a macrophage or a dendritic cell, which has engulfed a specific pathogen, takes fragments of the protein from that pathogen and puts it on the surface of that cell. A helper T cell that has the right antigen receptor that could recognize that molecule is now going to be activated. And part of that receptor is a protein called CD4, and that's where we get that name for that. So the helper T cell is going to produce cytokines, and it will both respond to its own cytokines, and it will activate itself even more. And the dendritic cell is also producing cytokines, which are going to activate it as well. So we end up with a clone of these activated helper T cells, because they're going to, once they become activated, B cells and T cells divide. And what they do is produce molecules, cytokines that activate B cells that are going to be producing antibodies, and also T cells, which are going to bind to and then consume by phagocytosis bacteria or viruses, whatever the antigen is for. So the helper T cells are a pivotal part of the immune response. And I'll show you this little video here. Okay, this little video, if you watch it again on your own, part of it is non-functional because it's like a little game. So only watch it to about a minute and 20 seconds, and then after that it starts to repeat itself.
Now let's look at how helper T cells aid cell-mediated immunity and humoral immunity. Imagine that bacteria have infected the body. Antigen molecules on their surfaces identify them as foreign. A macrophage engulfs an invader and breaks it into pieces. It displays the foreign antigen on its surface, along with a self protein that acts as a label for the body's own cells. The macrophage is now an antigen-presenting cell. The antigen-presenting cell secretes interleukin-1. Both interleukin-1 and contact with the antigen-presenting cells activate specific helper T cells to divide and grow into many cells with the same receptors. The helper T cells next activate other cells in the immune system by secreting another stimulatory protein, interleukin-2. Interleukin-2 stimulates helper T cells, cytotoxic T cells, and B cells. The helper T cells divide more rapidly and increase their stimulation of other cells. Cytotoxic T cells are stimulated to become active killer cells. B cells are stimulated to develop into plasma cells that secrete antibodies against the specific antigen.
So the humoral response is the secretion of antibodies. And It starts with the activation of B cells. Antibodies generally are not killing bacteria or destroying viruses. Instead, they tag it for destruction by other cells, the neutrophils and macrophages. Viruses can be neutralized by antibodies, especially if the antibody is binding to whatever protein on the viral surface the virus is using to bind to cells to get into the cells. And that's what they're talking about when they say we're looking for neutralizing antibodies against COVID. That means we don't just want antibodies that can bind to the virus. We want an antibody that's specifically going to bind to the spike protein so that the virus will not be able to enter cells. So neutralization means that the virus is prevented from binding to whatever it binds to get into the cell. And generally, viruses are binding to some normal surface protein on a cell. So if you can prevent that from happening, that's neutralization. That's the kind of antibody that we want. Opsonization, this is the tagging of a cell, like a bacteria, with an antibody so that macrophages will now come along and say, this is something that I need to get rid of, and will consume it then. So here's another little video on that. This is a pretty short one.
The main role of antibodies secreted by plasma cells is to tag foreign molecules or microbes for destruction. Binding of antibodies to antigens can trigger several effector mechanisms. Neutralization occurs when antibodies bind to antigens, blocking their activity and making them harmless. For example, antibodies cover the surface proteins of this virus, preventing it from hooking up with the host cell. Other antibodies may bind to toxin molecules on a bacterium. The bound antibodies attract macrophages, which dispose of the invaders by engulfing them, a process known as phagocytosis. Antibody molecules have at least two binding sites, allowing them to form a clump of microbes by binding to two antigens on two different cells. This process, called agglutination, traps microbes and makes it easy for macrophages to capture them and dispose of them by phagocytosis. Precipitation is similar to agglutination, except this process clumps together free antigen molecules dissolved in body fluids. This inactivates them and attracts macrophages, which destroy them by phagocytosis. One of the most important mechanisms of humoral immunity is the activation of complement proteins. Binding of antibodies to a foreign cell triggers complement proteins in body fluids to form a hole in the membrane of the target cell. The hole allows water and solutes to enter the cell. The foreign cell swells, bursts, and dies. Humoral immunity, mission accomplished.
So the humoral and the cell-mediated responses have both a primary and a secondary. The primary response is slow. The primary response takes about 2 weeks to reach maximal antibody production and maximal availability of cytotoxic T cells. The secondary response is going to be much stronger, and that's often why booster shots are given to get you up to that secondary response level so that you will actually completely prevent infection the next time you are exposed to it. The response gets greater the second time, and it's much faster the second time. So we have the humoral response, the antibody response on one side, cell-mediated on the other. Oh, wait, let me go back to my little pointer here. So here's our antibody, or our antigen exposure. First exposure to the virus or the bacteria, whatever it is. We have our innate response, which is macrophages, including dendritic cells, are going to consume this, whatever it is, virus or bacteria. They're going to take some of those proteins and put them on their own surface and then head off looking for T cells and B cells. The antigen presenting cell is going to activate cytotoxic T cells, which are going to clonally reproduce. It's going to activate helper T cells, which are going to clonally reproduce. And those helper T cells are going to activate B cells, which are going to clonally reproduce. So we have multiple kinds of cells here that are going to form memory cells. We have memory B cells that are going to be able to secrete antibodies and stimulate that second response that's going to be much higher. We have memory cytotoxic T cells, which when that antigen is presented again, are going to produce active cytotoxic T cells. And you'll notice here that our helper T cells are pivotal here in this response. And this is the cell that is eliminated by the HIV virus. Remember, these are the CD4 cells. And that is the protein that HIV binds to get into cells to reproduce. And then it kills those cells eventually. So when you eliminate these cells, you eliminate this whole activation process here. And that's the characteristic symptom of AIDS, is that response to many pathogens is just completely diminished because we are eliminating this one type of cell that's kind of eliminated, that's pivotal in this process. So here's one of those cells, a helper T cell, and we have the CD4 binding protein on the surface that just so happens to be the protein that HIV binds to. So viruses just stick to some protein on the surface, and which protein they stick to is going to determine which species that virus can infect, which cells in a species that virus is going to infect. A virus might be able to infect many different kinds of cells in the body, or it might only be able to infect a very tiny subset of cells in the body. So for example, cold viruses can bind to mucous membranes, but they can't bind to your liver cells or your brain cells. So they're never going to affect those organs. HIV can only bind to these helper T cells that have the CD4 epitope in their receptor. And eventually they're going to destroy those cells by hijacking the entire reproduction machinery of the cell into making more virus particles. I'm not going to show you this little video. This is a nice little review of all of this, but it's about 6 minutes long. So it's in the PowerPoint, so you can watch this little video on your own. But it is a nice little review. So immunization is giving you that primary and sometimes even a secondary exposure, if there's a booster shot, to some antigen. And it's generally not a live bacteria or an active virus, although sometimes it is. Sometimes it's what we call an attenuated virus, meaning that it's not capable of causing a severe infection. But for certain vaccines, like for the polio vaccine, the attenuated virus vaccine with the live virus, even though we know viruses aren't really alive, elicits a much better immune response than just the bits of the virus particle, which is in the killed vaccine. Although now that polio has been eliminated from the US population for decades, generally we don't use the live vaccine anymore because we just don't need to, because it's not a likely threat at this point. It's kind of the polio vaccine at this point is kind of a just in case kind of a vaccine. So the immunizations and vaccines are our best defense against viruses. We have very limited treatments against viruses. Vaccination is far better, and the vaccination programs have been super successful. We've even managed to eliminate one deadly virus completely from the human population, and that is smallpox. Nobody who was born after 1979 has needed to get a smallpox vaccine. I've been vaccinated against smallpox. It was required. There was no opting out. And smallpox has been completely eliminated. So nobody has had a case of smallpox in the world since 1979. It was declared extinct. Now, there are some frozen samples of smallpox, and there's some in the US and there are some in Russia. Um, and also the methods for making the vaccine is also still known, just in case. But hopefully polio is next to completely eliminate. And one of the things that's accomplished with vaccines that we used to have to accomplish only by exposing everybody to something is something called herd immunity. So herd immunity means the whole herd is safe, even though not everybody is immune. So if we have, and it varies by the virus, how contagious the virus is. For something that's super contagious like measles, we have to have 95% of the population immune in order to reach herd immunity. If it's something less contagious, like for the COVID-19 virus, they estimate that about 70% immunity would give us herd immunity against that virus. And why is that important? Well, not everyone can get vaccinated. So you can't just say, oh, every individual's choice to get vaccinated. No, some people can't be vaccinated. Kids who are undergoing cancer treatment can't be vaccinated. Often adults who have just been through chemotherapy or bone marrow transplant, they cannot be vaccinated. Some people have particular immune system disorders. They cannot be vaccinated. Infants can't get most vaccines until after age 1 or sometimes even later. So uh, by getting vaccinated, if everybody else who's healthy gets vaccinated, that protects all the vulnerable people who don't have the choice. They don't have the choice to get vaccinated, and that protects everybody else. As we found out the hard way with measles, because it's so contagious, if even a few percentage of the population stops vaccinating their children, suddenly we have an enormous outbreak. Polio has almost been eliminated. So here we have, this is several graphs here superimposed. So let's look at this. So on the y-axis here, we have deaths from polio in the United States, and then years down at the bottom here. So before the vaccine was introduced in the 1950s, several thousand kids a year, and I say kids because it was mostly kids, died, and that doesn't even, and there were at least 10 times that number who were permanently physically disabled from polio as well. Although many kids did recover completely from polio, but often it would be a multi-year illness, and even if they did make a complete recovery. So polio is nothing to wish on anybody. And then as soon as the vaccine is reduced, the number of cases diminishes rapidly until now. We haven't had polio in the United States for decades. And we see the comparison here between 1988, when the worldwide polio vaccine push really started. So purple are the countries that no longer have any cases of polio or any community transmission of polio. And by 2014, we were down to 3 countries that still were having community transmission of polio, and that's where we still are today. We're down to 3 countries that still have polio, and part of the reason is resistance to vaccines. People being suspicious of vaccines, thinking that the vaccines are actually going to poison their kids or something. But they have, the World Health Organization has now been recruiting people from the countries themselves to be the ones giving the vaccines, so that people will be less suspicious. But there had been a goal of totally eliminating polio by the year 2000. That didn't happen. The year 2010 didn't happen. And then by 2020, and well, now we have coronavirus, so it's not going to happen either. But hopefully in the next few years, we're going to completely eliminate polio from the human population. this is a similar table for measles. Measles is one of the most contagious viruses that we know of. It is quite easy for a single infected person to infect 200 other people. It's very infectious. It's airborne. It stays in the air for many hours. So you can walk into a room where an infected person was 5 or 6 hours after that person has left the room and still get the measles. So it's extremely contagious, very airborne. It's much more contagious than the COVID-19 virus. And it's also deadly. Some people recover from it. It's way more serious an illness than chickenpox. I had measles myself when I was 5, and I was extremely ill. I should have been in the hospital, but my mom was a pediatric nurse at the time, so she wanted to keep me at home because she knew one of the complications of measles in children is getting secondary infections, because measles lowers the effectiveness of your entire immune system. And she knew that many kids with measles ended up getting something else in the hospital and then dying from that. So she kept me at home, even though I was extremely ill and probably should have been on an IV. So it's a very serious illness. It's not any fun at all. It's not like chickenpox where you sit home and watch TV for 2 weeks. So recent anti-vax information, and you can see right here, you know what happened right here? This was when the initial surge of anti-vaccination information came out about autism and vaccines, and it turns out there is no connection whatsoever between any vaccine with mercury or without mercury and autism. That has been absolutely, totally disproven. The guy who, Dr. Wakefield, who first published that study, it's now been shown that he actually fabricated data for a lawsuit. He was paid by a group of lawyers to fabricate the data to win a lawsuit against a vaccine company. And it sparked this whole mess of anti-vaccine stuff, which has been going on for 30 years and is still going on today. Just started by greed. So a group of greedy lawyers and one greedy doctor have caused the death of thousands of people by all of their anti-vaccination misinformation. And I don't think I'm exaggerating at all when I say that. Prior to that, prior to 1987, I think when this study was published, this fake study, there was no anti-vaccination sentiment. Nobody was anti-vaccination. People were so grateful to have all these horrible, deadly diseases being prevented. Nobody was anti-vax before that. I know, it's hard to believe, but it's true. So active versus passive immunity. So active immunity is your own body responding to the virus. Active immunity is your own body making memory cells and making antibodies, and that passive immunity is short term and it is put into your body. And there's I don't know what that was. I'll find out later. Passive immunity is put into your body, and there's natural passive immunity, which is what you get when you're a fetus or a newborn. So babies get antibodies crossing the placenta and get some immunity at birth to whatever their mother was already immune to, and that continues after birth. If they're breastfeeding, antibodies, breast milk is full of antibodies, so whatever the mother is producing antibodies to, those antibodies are in the breast milk, and that will provide the breastfeeding baby with some immunity to whatever the mother's immune to. That's passive because the baby's body is not making it. Now, we do have some therapeutics that are passive immunity as well, one of which I already mentioned, the monoclonal antibodies that are going to be injected into somebody to give them temporary resistance against some particular virus or bacterial infection. Actually, that's what rabies treatment is. If you are bit by a rabid animal, because almost nobody gets a rabies shot, there is a human rabies vaccine, and people who work with wildlife or work with bats usually are required by their job to get a rabies shot like a dog would have. There's a human version of the rabies vaccine, but most people, most your average person, has not gotten a rabies vaccine. So if you are bitten by a bat or another animal that's suspected of possibly being rabid, the way they treat you is with IgG injections. They have antibodies against the rabies virus, and they inject you with it for several weeks to give you passive immunity against that virus so it can't infect and attack your immune system. And that's passive immunity. Now, this is also what that Regeneron treatment is for COVID. If they are giving you an injection that contains antibodies, that is passive immunity being used as a therapeutic. And it doesn't make you immune to it. It is temporary. Passive immunity is temporary. It wears off. Those antibodies will slowly be removed from your body. IgG antibodies have a half-life in the body of about 20 days. So that means in about 6 weeks, whatever passive immunity that you got from an injection or a treatment is going to be eliminated from your body. So, but we already use these. We already use antibody treatment for certain diseases, and it works. It works very well. The rabies treatment works very well, but you have to get it immediately. You can't wait a week and then go get the rabies treatment. You have to get it right away after you're bit. And immunity also plays a part in blood transfusions. Probably all of you know, or maybe you can remember from Bio 1151 when we talked about genetics. There are there's actually a lot of subcategories, but we have different antigens on our red blood cells. There's an A antigen, a B antigen, and if you have neither antigen, then you're type O. So that's why type O is recessive. The gene itself is actually for an enzyme, and so if you don't have an active version of the enzyme, you're not going to have either A or B antigen on the surface of your red blood cells, so that's O. And this is going to affect how people will react if they get a blood transfusion. So if your blood type is A, you won't have antibodies against A, but you will have antibodies against B. So we can't give you blood that has a B antigen in it. So we can't give you type B and we can't give you AB, but we could give you A or O. If your blood type is B, that means we can't give you blood with the B antigen in it. Or we can't give you blood with the A antigen in it because you'll have antibodies against it. You won't have antibodies against your own antigen. So if your blood type is B, you won't have anti-B antigens or antibodies. So O, people who are blood type O, will react to A or B. But O doesn't have the A or B antigen, so that's why O is the universal donor. And the rhesus factor positive and negative is the same. The positive, which will make your blood type positive, that is another antigen on the surface of the red blood cells. If your blood type is negative, that means you are simply lacking that antigen. So O negative doesn't have, if your blood type is O negative, you don't have the A, B, or rhesus positive antigens on the surface of your red blood cells, so your blood could be given to anybody, and they won't react to it except for the very rare types. There's a few very rare types that would even react to O negative, but they're less than 1% of the population. But this is an important concept for people who are going into the medical field. Autoimmune diseases are when the body's antibody systems and T cells now start responding to a self-protein. These cells were supposed to be either eliminated or inactivated. And there are quite a few diseases known, which we call autoimmune diseases. Auto means self. So autoimmune diseases are diseases in which the immune system reacts against a self-protein. And more and more are being discovered all the time. Diseases that have been known for a long time, like type 1 diabetes, have been found to be actually autoimmune diseases. So lupus, rheumatoid arthritis, type 1 diabetes, multiple sclerosis are examples of diseases that have been known for a long time, but have only more recently been found to be autoimmune diseases. I believe Crohn's disease and ulcerative colitis are also now autoimmune diseases. Um, and it's not clear why this happens. Sometimes it's thought, like with type 1 diabetes, that it's caused by exposure to a virus that has a similar epitope to the beta cells in the pancreas. But it's something that we need to do more research on, clearly. The immune system is affected by general health conditions in the body. Severe exercise is bad for the immune system. Moderate exercise has been shown to improve the function of your immune system. But people who over-exercise, it actually leads to more frequent infections and increases the risk of things like pneumonia. Psychological stress has also been shown to depress the immune system. People who have chronically high levels of cortisol have been shown to have lower natural killer cell activity, lower antibody response to vaccines. So stress is a big deal. Getting enough sleep is also really important for the immune system. People who, if you short sleep, and that means sleeping less than 6 hours. If you short sleep even one night, it detectably lowers the number of natural killer cells that you have and your antibody response. So people who get a vaccine after one night of short sleep, it lowers the amount of antibodies that are produced in response to the vaccine by about 50%. And that antibody production does not recover even if the person gets a good night of sleep on the next few subsequent nights, which is a really interesting study. So if you're planning on getting a flu shot, get enough sleep the night before. If you don't get enough sleep, get your flu shot on a different day, because the flu shot will not be as effective if you got 4 hours of sleep the night before you get a flu shot. You're better off waiting and getting it on another day. It's really interesting result of how important getting enough sleep is for the normal, healthy functioning of your immune system. And this could totally explain why, and this has been known for decades, that shift workers, people who don't get enough sleep, people who work night shift, people who change their shift all the time, have an increased risk of cancer. Because if you're reducing the function of your natural killer cells by not getting enough sleep, that's one of our primary defenses against cancer, progressing from a couple of abnormal cells to an actual cancer. So sleep is important, is the lesson from that. Besides autoimmune diseases, we also have diseases of immunodeficiency, and many of these are genetic and are present at birth, but they can also happen later in life, usually due to exposure to chemical agents. Sometimes it happens deliberately because the person has a white blood cell cancer, like some kind of leukemia, and the treatment for the leukemia gives them a permanent immunodeficiency. So the only way to fix the inborn immunodeficiencies is with some kind of gene therapy, which there have been a few successes with that. An acquired immune deficiency later, AIDS is an example of that as well. AIDS stands for acquired immunodeficiency syndrome, caused by the HIV virus, but it can also be caused by other kinds of exposures or even as part of a medical treatment. So we talked about innate immunity in this chapter, starting with barriers, mucous membranes, and then the immediate reaction of the body to the presence of bacteria or viruses that are not specific to a specific type of virus or epitope. Phagocytotic cells, peptides, and other chemicals that inactivate bacteria. And then we have the adaptive immunity. This is the antigen response. with the humoral response using B cells, the cell-mediated response using T cells, excuse me, uh, and then we have the memory cells, which allow the secondary response to the same virus or bacteria to be much higher. And that memory response is what vaccines do. So vaccines give you a non-risky exposure to a particular virus or a bacteria by exposing you to just a piece of it so that you will mount an immune response to it when you do see the real thing. Active immunity means the person's own body is doing it. Passive immunity means that some other body is giving it to you. So whether it's the mother through the placenta or through breast milk, or it's some kind of a therapeutic like monoclonal antibody therapy would be an example of passive immunity.